Hello, I'm Gaurav. I've done my PhD from India, and currently I'm a postdoctoral fellow in Lund University in Malmö, Sweden. And today I'm going to talk about the role of a methyltransferase in the beta cell function. So the methyltransferase that I'm working on is a dimethyl adenosine transferase 1, whose function in beta cell is not completely understood. So what we know about this DIMT1 is that it's a paralog of the TFMB1 mitochondrial transcription factor, and its functional role in beta cell is not understood. The DIMT1 is expressed in the islets and the clonal beta cells, and it is also regulated by the glucose. And the current DIMT1 sequences suggest that it is a methyltransferase that belongs to the methyltransferase superfamily, and it helps in the ribosomal RNA processing. So what we have observed that DIMT1 level is altered in the type 2 diabetic patients, and that's why we asked the question that what exactly happens in DIMT1 during this diabetic phenotype. So based on that, we build our hypothesis, which is that if we knock down the DIMT1, there will be no methylation of the 18S as ribosomal RNA, and followed by there will be attenuation in the mitochondrial protein translation, and that can perturb the mitochondrial functional parameter, which is oxygen consumption rate, adenosine triphosphate, and the mitochondrial membrane potential. And these parameters, which is very important for the mitochondrial function, and this can lead to the decrease in the beta cell stimulus secretion coupling, and that can lead to the depletion of the insulin secretion followed by the diabetes. So to do this, we have followed this methodology, and for that we knocked down the DIMT1 with the siRNA technology, and for that, we have used real-time PCR and the Western blot to validate the knockdown. After getting a validation of the standardization of the knockdown of the DIMT1, we proceed further to check the insulin secretion in the cells, and we checked it with the low glucose 1 millimolar and the high glucose 20 millimolar. For the mitochondrial function parameters, we took mitochondrial membrane potential and ATP-ADP ratio with the Perceval, and the mitochondrial electron transport chain systems protein expression with the help of Western blot. For the protein synthesis, we analyze the protein synthesis with the help of O-propygyl pyromycin. It's a click chemistry based Alexa fluor indicator of the protein synthesis, and we check the protein-protein interaction with the proximity ligation assay. For the methylation assay, we checked the primer extension assay and also the ribomet sequencing to see the methylation status of this DIMT1 in the endo-CB1 cells. So to begin with, as you can see, we knocked down the DIMT1 level in the endo-CB and BH1 cells, and we found that the 100 nanomolar of the siRNA is effective, and it knocked down 80% of our DIMT1 from the endo-C cells. And then after, we wanted to see whether DIMT1 methylates the 18S ribosomal RNA in the beta cells, in the endo-CBH1 cells. For that, we used the primer extension assay, and for that we took a special kind of enzyme which is known as the hemoclen Q enzyme, which gets interfere with the secondary structure or the any methylated structure, so it will not transcribe your mRNA copy. But when we knock down the DIMT1, as you can see from the 50 nanomolar and 100 nanomolar, there was an increase in the copy number of the DIMT1, suggesting that once the DIMT1 is knocked down, it can transcribe the mRNA. And for the positive control, we took revert aid, which transcribe your gene regardless of any secondary structure and the methylated structure in your system. Okay, then further we moved on to check the insulin secretion in the cells, and as you can see in the endo-C cells, we saw a significant increase in the insulin secretion at the 20 millimolar concentration of the glucose, whereas when we knocked down the DIMT1, we found there was a significant decrease in the insulin secretion, suggesting that DIMT1 play a very significant role in insulin secretion in the pancreatic beta cells. We then moved on, and then since we knew that DIMT1 has a role in the beta cell translation, so it was very imperative to check the level of this protein synthesis in our system. So for that, we checked the protein synthesis with the help of a puromycin based assay, and which is a click chemistry based assay, and it is stacked with Alexa floor. So the puromycin bind with the nascent protein, so the more the protein is translated, the more the OPP will bind on that, and then with the help of the fluorescence we get the level of the protein synthesis in the cells. So as you can see that in the scrambled cells, when there is no OPP, we hardly have any fluorescence, but as soon as we added the OPP, the level of the protein synthesis significantly went up, and when we knocked down the DIMT1, you can see there was a remarkable decrease in the protein synthesis. And for the positive control, we use the cyclohexamide, which completely blocks the protein synthesis. So these results suggested us that the DIMT1 has a functional relevance in the beta cells, including the insulin secretion and the protein synthesis. Now we moved further, and we wanted to check the role of this DIMT1 in the mitochondrial function, because as you know, the mitochondria is very important for the insulin secretion in the beta cell. So we thought that what would happen to the mitochondrial function if we knock down the DIMT1 from the cells. So for that, we chose 4 critical parameters of the mitochondrial function. The first one was the oxygen consumption rate, and we saw that there was a maximum respiration capacity was observed in the cells without any DIMT1 knockdown. So these blue lines are the scrambled cells; these are the control cells. But when you see these orange lines, which is the DIMT1 knockdown cells, you can see there was a blunt response of these cells, suggested that the DIMT1 has affected the overall oxygen consumption rate in the endoc cells. To move further, we also checked the protein expression of the electron transport chain system of the mitochondria, and we saw that there was a remarkable decrease in the level of these complexes in the mitochondria. As you can see, C5, C3, C2, C4, this was all down-regulated at the DIMT1 knockdown cells. To get the results from these complexes, since you can see the C5, which is ATP synthase, we were curious to check does DIMT1 affect the level of the ATP, because ATP is required for the secretion of the insulin in the beta cells. And what we found that with the help of Perceval, which is a GFP indicator of the ATP, we transfected the cells with the Perceval, and we found that in the controlled cells there was elevated level of the Perceval signal, suggesting the physiological level of these ATP in the cells, whereas in the DIMT1 knockdown cells, we saw a very diminished level of the Perceval, suggesting that DIMT1 has an effect in the overall ATP production of the cells. And finally, we checked the mitochondrial membrane potential. As you can see with this data, that we used TMRM for the mitochondrial membrane potential. As you know, that TMRM passively goes inside the mitochondria, and it fluoresces inside the confocal microscopy, so you can analyze the level of the membrane potential. And what I saw that there was a decreased mitochondrial membrane potential in the DIMT1 knockdown cells as compared to the control cells, suggesting that DIMT1 has an effect on the mitochondrial membrane potential. So overall, what we have witnessed that DIMT1 methylation is very important for the cellular function, especially in the mitochondrial translation and the beta cell function. So what we have shown till now that the DIMT1 methylation is important for the beta cell translation, as it is involved in the stabilization of the 40S ribosomal subunit, which is a small subunit of the eukaryotes. And when you knock down the DIMT1, there is no methylation in these 40S subunits, and that leads to the destabilization of this 40S subunit, which could probably interfere with the interaction of the big and the large subunit of this ribosomal subunit to bind with it. So this is our developing hypothesis that we have put forth, that in the control cells, as you can see, the 60S and 40S binds after the stabilization process, and they translate the protein. So you have high protein synthesis, you have normal insulin secretion, you have OX-phos expression, and you have healthy mitochondrial membrane potential. Whereas when we knock down the DIMT one here, as you can see, when you knock down the DIMT one, there is no methylation, and that methylation is very instrumental for the ribosomal subunit stabilization. So you can see there could be a probable destabilization of this subunit that can interfere with the interaction of the 40S and the 60S subunit, and that could lead to the perturbation of the protein synthesis, and we can have the final phenotype like decreased protein synthesis, decrease in the insulin secretion. We can have altered OX-phos expression and the dissipated membrane potential that we have observed in our result section. To work in the future, we are looking to understand the role of this DIMT1 in ribosomal assembly. So we are currently investigating the role of this DIMT1 in ribosomal assembly by the proximity ligation assay, and we are also trying to investigate the target of this DIMT1 in these beta cells with the help of the RiboMeth sequencing from the Illumina platform. So overall, the results are very interesting, and what we have shown that this DIMT1 methyltransferase activity is very critical to the beta cell translation. As of now, we have not identified the mechanism behind it, but we are currently investigating the targets of DIMT1 that could possibly open a new avenues to target the diabetes and the therapeutics in the future. I would like to thank my postdoc mentor, Professor Henrik Mulder from the Lund University. I would also like to thank the Molecular Metabolism Unit in Lund University, who has always helped in giving the significant inputs to progress in my project. I would also like to thank the EASD for giving me the travel grant to attend this conference and to present my work. Thank you so much.